Okay, so now we have Ann West, who you've heard of, has been up on the stage before, but now we're going to hear what we promised earlier. Ann? Okay, let's sit back down. I'm sorry, who told you to sit up?
Hello, I'm Ann West, and I'm going to be presenting on my family's DNA sequencing as well as genotyping data and the medical, scientific, educational, and ethical perspectives from this. First, I'd like to thank the organizers of the GET conference for letting me speak here, the Illumina Personal Genome Service for providing my family with DNA sequencing data. Before them, 23andMe for providing the four of us with genotyping data, my biology teacher, Dr. Blickenstaff, and my parents for helping me by letting me bounce ideas off of them, and several public data sources listed here that I used. I'd like to start by going over my family's genetic background. We have several diseases in the family that may or may not, but most likely do, have some genetic component. And they are cancer, heart disease, Alzheimer's disease, deep vein thrombosis, and diabetes. Now, starting off, this may seem like a rather large number of diseases, but it's not an unusual number, actually, because it includes my entire extended family, and that's a lot of people. However, though my grandparents have participated in the 23andMe testing, I have not so far used their data, so the only data that I have used from my family is the data from all four immediate family members, myself, my brother, and of course my parents. I'd like to focus first on deep vein thrombosis. I decided to analyze the factor V gene first, seeing as the factor V Leiden mutation is readily identifiable as the mutation that causes deep vein thrombosis, whereas the origins of the other diseases remain genetically ambiguous. The mutation of the factor V gene causes deep vein thrombosis. Clots often form unnecessarily in the legs and can break loose and go to capillaries in pretty much anywhere in the body. If it goes to the heart, it causes a heart attack, it can cause a pulmonary embolism in the lungs, or potentially a stroke in the brain. So it's very dangerous if untreated. However, if diagnosed, there is a straightforward preventative treatment that one can do. Coumadin is off-patent and inexpensive medicine. It's an anticoagulant drug. You need to be a little bit careful with your lifestyle, particularly making sure to get up, stretch, and drink lots of water whenever you go on long flights. And be careful with drug interactions, such as with birth control pills. The risks that the actual gene itself cause: a heterozygous mutation, or having one tainted copy of the gene, causes 2.7 times relative embolism risk, and if you're homozygous for the factor V Leiden mutation, you have an 18 times higher risk of an embolism. Literature cites numerous clinical cases of what is known as compound heterozygosity, or having multiple genes in the mutations in the factor V gene, and having one of these be on one gene and the other be on the other gene, so that though you're heterozygous for both, you have two differently mutated copies of a gene. If you have only one mutated gene, then the other is still making normal protein, but if you have two mutated copies, then both are making defective protein, so the phasing of mutation location can actually make a big difference in clinical outcome. I'd like to look here at the recombination rate in the factor V gene with data provided by the International HapMap project. What we have on top here is a graph of recombination rates from the HapMap project against their position on the chromosome, lined up with two green lines above the UCSC genome browser representation of the factor V gene region located on chromosome 1. The places where recombination rate data spikes are crossover hotspots, and the rest of the places where recombination rate is next to 0 are crossover cold zones. So the gene is not one haplogroup, so it's hard to tell if you have multiple mutations in this gene, if they will be inherited together or separately in the data. And furthermore, it is hard to determine this if you have compound heterozygosity or not from the data from one individual. Here, experimental data confirms the location of a crossover hotspot at the longer green line. What we have here are a series of minor allele frequencies from the 1000 Genomes project. So the bottom ones are recently occurring mutations in the gene, but some of the ones on top accumulate together, and there are boxes around them in red. As you can see, the frequencies change at the lines, thus giving evidence of a recombination hotspot there. So I decided, in order to determine compound heterozygosity and chromosomal location of various mutations, I decided to use the data that comes from a whole family. What we have here is a picture of me with the 23andMe sample collecting kit. My family had their genotyping done in 2007, and the picture on the right is of the chromosome, is a diagram of the chromosomes of my SNP alleles that were tested by 23andMe compared to those of my brother. The dark blue regions represent where we are completely identical in our SNP alleles, the light blue where we are half identical, and the white where we are not identical at all. Changes between any of these colors indicate that there was a crossover in that particular region. Gray is where there was not enough data to determine this. Oh, and I tried to locate crossover points using this graphic representation. However, the data is fairly crude just from this representation, seeing as a couple of hot crossovers were missed, and furthermore, by measuring along this picture, which was time-consuming and tedious enough as it was, I was only able to locate the crossover points to about within 1,000 bases of where they actually were. Sorry, 1,000,000. So I decided to use a feature of the 23andMe website, which is the raw data download. You download all of your data in a file that comes in TextEdit, very long file, for each person, and this gave me access to over 570,000 SNP loci for each person, over 100,000 of which were informative for the location of paternal and maternal crossover points. Looking at one of the earliest SNPs on chromosome 1, I identified a maternal phasing point, not necessarily a crossover, where my mother is heterozygous AG, and at the same location, my father is homozygous GG. My brother and I both have the genotype AG. So we know that we both inherited a G from our father, though we're not sure which one it is, and we know that we both inherited the same allele on the maternal side, the A, from the same maternal grandparents. However, this was also inefficient, so I decided to analyze the data to look for crossover points in Microsoft Excel. Here, I located crossovers on the maternal side. I took SNPs where my mother was heterozygous and my father was homozygous, and used this to determine if in my family, if my brother and I had the same maternal allele or different maternal alleles. The places where it goes up are where we are the same. The places where it goes down are where we are different. The changes are sites of meiotic recombination, and on the bottom with the red bracket, I marked where the factor V gene is located. I did the same thing on the paternal side, and as you can see, there are no crossovers in the factor V gene region here either, despite the fact that it straddles a recombination hotspot. I then decided to use the 23andMe data to locate regions where crossovers had occurred. This is one of them in the red line, and the blue line on the graph is of recombination rate data from the International HapMap project. Though you can tell that the crossover region overlaps a crossover hotspot, you cannot tell if it actually lands on the crossover hotspot or falls on the crossover cold zone. Its location is ambiguous. So, would sequencing give me any more precision in this? This slide is of the process my family had to go through for sequencing, from the consent forms and writing up of a prescription, since we used Illumina, having our blood drawn at El Camino Hospital. Here's the initial sequencer spot image, the initial data from the sequencer, which is rather hard to read, so it was simplified into sequencer reads on the bottom right for us. And the picture next to that is a close-up of the aligned reads, and next to that is an allele table of the SNPs, or any place where people in my family differ from what is considered to be the human genome reference sequence. So the answer to my question was yes, sequencing data does give me more precision when locating crossover points. In red, I located a crossover point using the 23andMe data, and in green, I used the sequencing data from my family to determine if that crossover could be narrowed down, and it was narrowed down to, against the HapMap data, a recombination hotspot. Returning to the medical analysis, I decided to analyze the factor V gene for compound heterozygosity. At first, I had about 1,000 SNPs from my family's sequencing data. However, I narrowed it down to these 14 on the slide here via crossing out or eliminating all loci that were not in the exons or within 50 bases of either side of the exons. The table indicates which alleles my father had and which ones he gave to me and to my brother, which alleles my mother had and which ones she gave to my brother and to me. In color are the missense mutations, or mutations that actually caused a change in the amino acid sequence coded for by the gene. The ones in green, I looked up first on dbSNP and subsequently NCBI and polyphen. Judging by their common rate of occurrence, which is over 20%, they are most likely benign, and polyphen also predicted these to be benign. In red is the factor V Leiden mutation, which is actually deleterious. As you can see, my father had one copy, which was passed down to me. So I am a compound heterozygote for my mother's mutations in green and my father's mutation in red. Fortunately, I already know that the mutations highlighted in green are harmless, so my medical result for the analysis of this gene is relief from my family that we only have one copy of the mutation of the factor V gene, which is the factor V Leiden syndrome being relatively treatable. Here I've listed some next steps, or what I plan to do in the future regarding my project. It took a while to analyze the factor V gene. So I plan to obtain some sort of software in order to do the heavy lifting DNA data sorting for me. I plan to discuss my results with a medical geneticist after analyzing other genes linked in factor V activity in the chart on the right, printed out from Wikipedia, and analyze the factor V promoter and other control regions as well. And yes, on the side, it would be nice to finish 11th grade as well. As for educational perspectives, this has been a chance for me to have a real-world application for conducting a sustained research project and to apply what I've studied in school. I still have a lot to learn, and bioinformatics is not well covered in my school's curriculum. But I've learned a lot. This has been great for my education, and I'm actually able to teach others about this too, my other family members and sometimes my biology teacher. As for the obtaining and preparing of original data and data samples, I plan to work in an actual laboratory setting this summer to gain hands-on experience with how samples are prepared and processed to produce data. The project itself that I've been conducting so far has involved laboratory work that has been done by other people. Here I've included a couple of slides of risk assessment. However, this was covered during the morning session, so if you have any questions, feel free to ask. And to sum up, I'm just beginning to analyze my data. It's time-consuming. Software to analyze the data beyond Excel would be extremely useful, so my father and I are actually shopping around for that right now. We already have a new scientific and medical result, however. We've aligned crossover locations with known hotspots 10 times more precisely, and our medical result is detailed characterization of the factor V gene region, including rare mutations and phasing of compound heterozygosity. We also have the relief that our known factor V line mutation is not 1/2 of a clinically significant compound heterozygote. This has been a great educational opportunity, and my family remains optimistic about the risk-reward trade-off that we have played in pioneering family genome sequencing. Thank you for your attention.
Hey, Ann, except there are going to be questions. So I know if we had to choose which family would be up for kind of dealing with this information, I think you guys have proven yourself the right family. I do not envy that biology teacher, though. So questions. We have time for a couple questions for Ann. Yes.
So, Ann, that was fantastic. Thank you so much. And by the way, about the 11th grade thing, screw it. Go get a job. But on a more serious note, so obviously you have this wonderful data set from 23andMe from the microarrays, and you also have this wonderful data set from the complete sequencing, something many fewer people are privileged to have. Have you done a compare and contrast? Have you looked at any cases where the two disagree, where one says that you should be concerned about something and the other says something different?
Actually, 23andMe has a remarkably low error rate. So when I did first obtain the 23andMe data, I sorted through it for Mendelian errors and errors where my brother and I would change from different to similar and then back to different on the maternal or paternal side for one allele, and that would be it. So those were probably errors, and I deleted them too. There are probably several others, but the error rate for 23andMe is less than 1%. So actually, in terms of clinical diagnoses, it's fairly accurate.
Anyone else? No? All right. Well, thank you so much, Ann. It was really wonderful.